get going. This is what I'm going to talk about, and it's going to be a bit of a speed run. I'm going to talk very briefly about the lead generation. I'm obviously speaking within the ASAP team, which has descended from the COVID moonshot. So there'll be many people who know a lot about that. I'm going to pick up target product, target candidate profiles, and then I'm going to look at risk appraisal of our lead series. And again, there's some really interesting tags into the previous talk. And I'm going to show you actually how we solved some problems, how we shortlisted to candidates. And then there's a very brief step into what we're doing in terms of pandemic preparedness in the ASAP program, and how this candidate then inspired some of the things we did next and some very early data, because it's really good to see. So I couldn't resist showing them. Okay, I suspect a lot of this audience are familiar with the role that proteases play, and in particular the main protease for coronaviruses. So this is a picture of the main protease. I don't think that we're going to, sorry, everything's jammed. Okay. That's better. Right, I got the slides advanced, and that was a horrible moment. So the main protease is responsible for cleaving the polypeptide, generating the proteins that the virus needs for replication. So if you block the protease, you block proteolysis, you block replication. As a medicinal chemist, that's what I need to know. I appreciate there's a huge amount more complexity in that, but for me, that's a good start. So in 2020, in the throes of the early stage of the pandemic, absolutely outstanding work done by the folks at Diamond and Oxford. So this was Martin Walsh near London, the Weitzman and Frank's team, particularly with Darren Ferron. So not only cloning, producing the SARS-CoV-2 main protease, generating atomic resolution crystal structures, running covalent screens, running fragment screens, and in just days over a month, publishing 70 fragment-bound crystal structures and releasing that into the public domain. As an inspiration of, so what could we do with that in terms of discovering drugs that might be protease inhibitors? And so the COVID Moonshot program was born. This was published last year in Science. The details of it are quite incredible. Multi-institute collaboration, crowdsourcing from hundreds of scientists contributing their designs into the public domain. So we had over 18,000 domains, which were then ranked, sorted, filtered. We actually made probably now just over 2,500 compounds finally made within the COVID Moonshot, using a variety of high throughput computational and experimental methodologies, hundreds of main protease structures solved, tens of thousands of measurements made, and in the end, a really attractive non-peptidic, non-covalent, completely new class of protease inhibitor came out of that. Potent at the enzyme levels, 30 nanomolar, potent at the antiviral level, so 64 nanomolar EC50, and with an acceptable rodent half-life certain for a lead. And all of this data was put into the public domain as it was generated. And it was a really exciting project to work on because of the speed it went at and just the international character of it. Now I'm going to step back a bit and talk about, again, that patient-centric approach that we've already talked about a bit today. And I think it's absolutely vital to think in this way. So we now know a huge amount more about the biology of SARS-CoV-2. Probably most of us on this call know about the pre-symptomatic phase, and then you get this rapid phase of extremely high viral replication. And in probably the majority of patients, even pre-vaccination, you would see that begin to drop down. But unfortunately, as we all know, very much to our costs, in a very significant number, you get mild to moderate illness, which is horrible. And then in a small proportion, people progress through into acute respiratory distress, and then unfortunately, critical illness in the ICU and far too many deaths. But at this stage, the virus isn't actually driving the disease. This is the stage where the virus drives the disease. This is an inflammatory disease. This is a viral disease. So this is our window of opportunity for an oral antiviral. And just those characteristics of the viral biology mean that inevitably you want an oral drug because you want to be treating here outside the hospital, which means you're looking at an oral drug. And again, mirroring what we've already been talking about in terms of target product profiles and target candidate profiles, this is what we drew up at the very start of the program as our best guess. And it's by no means as thorough. It's pulling out just critical features we knew we needed. And I'm going to highlight just 3 of those. So this PK-PD relationship, so continuous free cover over viral plaque formation for 24 hours. We want low DDI, so low drug-drug interactions, because obviously you have patients with comorbidities or maybe want combination therapy or are just taking other medications in their lives and you don't want them to stop. And clearly, our patient group has a significant proportion of women of childbearing age. So the whole feature around mutagenicity and heterogeneity is absolutely key. So those are 3 points that I've just pulled out here as being very important we want to focus on as we go from what is a lead and we're trying to turn that into a drug. So what are the issues? When we look at a lead like this, we take a kind of fairly dispassionate view of it. Well, there's a potential issue about plasmization. We have this central amide. Our in vitro and in vivo clearance really is a bit high. It's not really what we want. But looking at it, the obvious toxicity risks of this are low. We have some principles for how we want to optimize compounds. So we want to keep our compounds small. And by keeping them small, that gives less opportunity for off-target risks. We want to keep within the substrate envelope, because if you're just binding to the area where the peptide substrate binds, any mutation of the enzyme to evade your inhibitor will also reduce substrate binding, and that would be a fitness cost. So therefore, you essentially build in a resistance to resistance. We've talked a little already today. We've spoken about cost of goods. Try and keep the compounds simple. Now, in our case, we wanted to avoid peptidomimetics. And that's because we wanted to present a different set of development and toxicity risk profiles to what we knew would be a lot of other people working basically from peptidomimetic start points. And again, we had a piece of thinking that was around do more covalent compounds. And we had many colleagues on the team who'd worked on covalent inhibitors, taken them into the clinic. And their view was, get potency first, and specificity, covalency later if you need it. Now, in our case, this series we took all the way through without actually having a covalent warhead on it, because we were able to get what we think is sufficient potency. And again, in this case, because we were focused on SARS-CoV-2 for a first-generation inhibitor, we were going for SARS-CoV-2 activity rather than broad spectrum. But right at the end, I'll come on to how we can then change that. So in terms of that first question about toxicity, it's a protease inhibitor, small molecules. This was one of our cores. How do they perform when you look at them against a whole rack of human proteases? And the answer is compounds are extremely clean. So we have very selective viral protease inhibitors. However, the Ames risk was then flagged up. So Matt, who was working on the program at this point from Postera, on a literature deep dive, found that this amino isoquinoline buried in this paper had shown up to be Ames positive. And that's a real problem. So Ames is not something you want to encounter as a drug hunting team, and certainly as a medicinal chemist. And in this case, it's absolutely not. It's not something we can gloss over. Our TPP is for use in the general population. So any mutagenicity or teratogenicity are not acceptable. But it's a problematic assay. It's notorious for being sensitive to oxidative impurities. So you've got to ask the question, is this literature real? So you've got to reproduce your literature data, and your compounds must be highly purified to be confident in the result. And finally, what are we going to do if it is true? So we could substitute that heterocycle, we could replace it, or could we lock it into the structure so it can't be released and is not needing to be used in synthesis, because use of an Ames positive aniline in synthesis creates a whole set of extra problems for you down the line. So we didn't want to go down that route as well. So here's the critical aims experiment we're going to set up. So there's the 4-amino isoquinoline ready to go. We did some quantum mechanical calculations for prediction. We are expecting it. It's been in the literature shown to be positive. If it's negative, which is possible, we would obviously have to take advice, but then we would just move ahead. We made some key analogues, which QM calculations suggested would be less likely to be Ames positive. So obviously, if those are negative, we could potentially move forward. And we also looked at this kind of full-sized compound because we also wanted to bear in mind that there is always the possibility that the Ames activity could have been due to intercalation rather than the amino isoquinoline. But there's also the risk that this could just be getting cleaved. And if they're all positive, well, we have got some backup compounds coming along. So this was where we were. We set off the Ames experiment. And because we wanted to be very careful with it, we actually conducted the experiment at 2 different suppliers. In the presence and absence of this S9 metabolizing mix, and we did it in multiple strains. So we will be confident that if we were going to pick up any Ames activity, we would, and we did, which was upsetting, I think is a good way of putting it. And not only did we see it, not only was the literature result confirmed, but our fallback compounds were also Ames positive, and it looks like the full-size compound as well. So this was obviously very worrying. We needed to have an approach to solving this problem. So what do we know about making substituents on this isoquinoline? Well, we can put substituents in here, and all of these compounds come from one series, so we're only changing this group. This is the enzyme potency against the protease, and compared to the 360 nanomolar, which is a basic compound, certainly if you add various groups, you can get a little bit more potency. You can lose a bit as you add electron withdrawing groups. But it's generally associated with the hydrogen potencies associated with the hydrogen bond basicity of this nitrogen, which is also associated with electron donation, which is also associated with ease of oxidation, which is believed to be the pathway that leads to mutagenicity. What about if we replace that whole ring? So here's the amino isoquinoline. What about putting in isosteres? And unfortunately, the vast majority of these are far less potent. The only one that showed basically equivalent potency is at very high risk of re-aromatization. So this, we were concerned, would be very readily oxidized back to the amino isoquinoline, and therefore will be this is a kind of Pyrrhic victory. So that's not great. So next option is, what about capturing the amino isoquinoline? And very early on in the project, we'd actually looked at trying to form cyclic structures around here. So we'd made this idantoin, which causes a bit of a drop in potency, but it's not huge. Slightly more reassuringly, you don't need this nitrogen. So you can just make the imide and you lose about 4-fold. But if you go all the way to the lactam, we're still losing some potency, but now there is no chance that this can release the amino isoquinoline. And reassuringly, how much potency you lose actually does depend on the side chain. So with a slightly more potent side chain, you only lose fractional potency by cyclizing it up. So this looks like a possible solution in that we can maintain potency and we don't release the amino isoquinoline. So what happens if we now put these into an Ames test? And this was extremely gratifying that in both of these compounds, where you have different side chains, but with the same spirolactamization, so potent compounds in the protease, absolutely no trace of Ames activity. You can actually get a clean Ames compound just by blocking this up. So the hypothesis that looks like if this isn't cleaved, you might manage to make the compound clean. So although that's a theoretical possibility, we didn't want to go down that route because that would still leave the possibility of generating the aminoisoquinoline either in situ or in in synthesis or in trace amounts, and that was not a route we wanted to go down. And we weren't able to. We made even more electron poor aminoisoquinolines, and we were still unable to remove the Ames activity. So it looks like we can make some good progress on this. We can evade our Ames problem by tying the compounds up. I'll just move on from that. So we've managed our tox risks, but our clearance is still too high. Now, there's a number of ways you can tackle a high metabolism. You can take a reductionist approach. Because this was during a pandemic, we opted to go for a kind of slightly faster approach. So we used in vivo cassette dosing as a very pragmatic solution. Identified a number of areas around the molecule where we thought there was a reasonable probability that we were getting metabolism, and set to work testing out each of those to see if we could block metabolism. Now, first of all, what about our spirolactamization idea? So you do get, on average, so these are multiple match pairs where we've taken different substituents and we've just cyclized around. So you do get, on average, not just in one compound, you do get a drop-off in potency, but very reassuringly, across 10 match pairs, we do get a drop in clearance, and that is a rat IV cassette. So this is in vivo clearance drops when you block this. So that looks like it's a definite winning strategy, which is good because we've just built it in to avoid our Ames problem. We looked at aldehyde oxidase, not a substrate. If you look at benzylic oxidation, again, this is very gratifying. It looks, when you put in the amide here, you actually improve potency and you decrease rat liver microsome clearance, again, match pairs of compounds with and without the carbonyl. So in the end, that looks like it could be a winning strategy. Substitution on the isoquinoline actually transpires that adding fluoros and fluoros not only are less potent, but they are more rapidly metabolized. So if somebody tells you that just adding a fluorine will solve your metabolism problems, the answer is, well, definitely not always. So this was not a winning approach for us. So we worked around those. What about looking at this side chain? So it's a glycine, you'd expect this to be cleaved. What about tackling that? So this is really interesting. Just simple substitutions on the glycine give really radically different effects on potency. So the CH2 versus the cyclopropyl versus the gemdimethyl, you see this huge change in potency. But in this case, you can maintain potency. In fact, in the cell, it actually slightly improves, and the clearance is reduced. So this is rat liver microsome by blocking here. There's a moment of medicinal chemistry geekery here. It actually transpires that the gemdimethyl places the carbonyl in a different place. That's the gemdimethyl compared to the cyclopropyl. So these polar plots show how actually this is probably placing the carbonyl where you want it, whereas this puts it in the opposite place. But the important bit from our perspective is we keep the potency and we reduce the clearance. So we've managed to make improvements all the way around the molecule. There were places, however, where we weren't able to make much progress. So we know that ring replacement is hard. Ortho substitution here was very hard. So you can't do much about N-oxidation if that's occurring. So this is what these spirolactams look like, actually bound in the SARS-CoV-2 main protease active site. So this is a crystal structure, not a model. I'll just rock it for a minute so that you get a picture of how well it's buried in there. What you see is this isoquinoline is binding deep into the P1, forming a hydrogen bond to the histine 163. There's no cysteine, no interaction with cysteine 145, which is the catalytic cysteine. The aryl chloride back here is really deep down into P2, and the lactam carbonyl is bonding to glue 166. So that all makes sense. The improved potency here, honestly, I don't think we can give a good structural explanation for that, but it's very clear from the SAR that these really help. So we just live with that. In terms of clearance, rat liver microsome clearance correlates really well with unbound rat clearance, which is very reassuring. So it suggests the majority of clearance is probably hepatic. If you look at cross species in terms of microsome, human, dog, mouse, and rat are all highly correlated. Again, giving, oh, sorry, this is millipig, giving good support that we should be able to access these as preclinical species for toxicology studies. And again, giving reassurance that human PK prediction based on metabolic scaling is probably justified. Back to that critical parameter that we've all talked about, so the free cover over the EC90. So here are 3 compounds that we shortlisted. All of them show very similar rat PK when corrected for protein binding, and in rat shows similar cover over the EC. This is A549 EC90. So here's overall leaderboard for these compounds. As you look at all of their properties, so the combination of the potency properties here, and then the pharmacokinetic properties, and finally safety properties here, compounds also tested against 29 target urethins panel, all really very clean. But there are differences. So in our shortlist, this compound on the far right is probably a bit more potent as a cellular antiviral. It's certainly more stable in microsome assays. And when we did the predicted dose to human, this was the one that came out with distinctly the best or lowest predicted dose. And so that was the compound that was nominated as DNDi 6510. If you then take that into SARS-CoV-2 infected mice, so these are really interesting mice that have had 32 enzymes knocked out, mouse enzymes knocked out, and 8 human metabolizing mice knocked, 8 metabolizing human enzymes knocked in. So it's a really good model for use for human disease. When you have enough cover, so if we look at this gray line here, that's the 8 hum mouse levels from a 50 mg per kg dose, and even at 50 mg per kg, you see significant reduction in viremia, but at 300 mg per kg, you see very extremely significant drop in viremia. But that is at 300 mg per kg, but that is still projected forward to be an acceptable human dose. In terms of drug discovery route, it's interesting to see you get less than 10 steps. Well, the discovery route is 8 steps. There's some quite interesting chemistry here that I won't dive into, but there's some really good work done by our colleagues, both at Enamine and also our process chemistry group. That route has now been improved and scaled, and we've delivered over 10 kilos of material. As I said at the very start, COVID moonshot team is spread completely across the world. The sun never set on the COVID moonshot, quite literally. We had incoming contributions and financial contributions from a huge number of organizations. Very grateful to all of those listed and so many more who made this possible, and all of our colleagues who worked across the world in delivering this. Has to be a particular mention to our colleagues, the Enamine chemists in Kyiv who worked on this, who despite obviously the hostilities, basically have worked completely through the war and continue to deliver compounds for the ASAP discovery program. And I'm hugely grateful to all of them for going completely above and beyond what any of the rest of us have to put up with. The project, although the COVID moonshot delivered a first development compound, obviously the ASAP program, as many of those on the call know, carries on. And one of the descendant compounds from that, so that we carried on working on that lead series, with the intent of expanding the spectrum. So that compound I've shown you is a SARS-CoV-2 inhibitor. We've got early data from in vivo studies with a now SARS-CoV-2 plus MERS inhibitor, and we see quite dramatic effects on reductions of in vivo viremia with one of our lead compounds. So that's extremely reassuring. And again, I'd just like to emphasize, these are non-peptidomimetic, non-covalent compounds with what we hope will be a really good safety profile. Okay, take-home messages. We have a new class of non-covalent MPRO inhibitors. They're effective in vivo, certainly as a series and as a class against multiple coronaviruses. High throughput fragment-based lead generation and computational methods combined with modern medicinal chemistry can deliver new chemical matter. We did this all under severe time pressure, resource pressure, because we're very focused, even in a distributed project, and an open science international consortium can deliver. And we're now using the same platform to prepare drugs for pandemic preparedness. Thank you very much indeed. I think I've probably run right to time, but I'm happy to take any questions.